good time. Welcome all to our Common Ground program tonight. We're very excited to have you here this evening with us. My name is Rick Davis. I'm with Conservation Nebraska's Common Ground program. I'll be your host, and um, I ask if you have any questions, Q&A session in chat, and we'll be able to ask those questions after the speakers tonight. We also have a very short survey right at the end of the webinar, so please take that. We'll remind you again with that. With us tonight, we have Dr. Daniel Snow. He has been with the Water Services Lab, Water Research Lab since 1990 at UNL and oversees all aspects of day-to-day operations there. We have Dr. Tania Biswas with us. She's the research lab manager for the Water Sciences Lab. We also have Alison Klein. She's from Kearney High Science Department, and she teaches chemistry, AP Chem, and physics at the high school. And she's been with Kearney High since 2015. First, we'll have Dr. Daniel Snow, then we'll have Alison Klein, and then we'll have Jesse Stardla and Tanya give us a tour of the research. All right, turn it over to you, Dr. Snow.
Well, thank you, Eric. I appreciate the introduction, and I'm really happy to be presenting some of the things that we're interested in at the university with respect to water resources in Nebraska. I'm going to kick off the presentations tonight with a short talk on groundwater in the Vadose Zone. So let me go ahead and share my slides. All right, so I'm going to just kind of do a brief introduction on groundwater and the importance of studying the Vadose Zone in Nebraska. Well, nitrate, if you didn't already hear about it, nitrate is one of our most common contaminants in our groundwater. And one question that I'm asked often is, where does it come from and what can we do about it? So hopefully this short presentation on the Vadose Zone will help answer some of those questions. Nitrate contamination has been going on for several decades now. There are many public water systems in Nebraska that are affected by rising nitrate concentrations. This map from the Nebraska Water Quality Report shows all of the systems that are now required to treat their source water for nitrate, and several that have administrative orders, meaning that they are very close to being in violation of the Safe Drinking Water Act, and others that are doing quarterly sampling. And what that means is they're getting close to this maximum contaminant level for of 10 parts per million nitrate in their well water. And it isn't just nitrate that they're dealing with. There's other contaminants like uranium. This map shows public water systems that are affected by increasing uranium levels. We now think that nitrate and uranium are related. So there's something going on with nitrate, and it's likely that something is also affecting uranium concentrations in our public water supplies. So where is this Vadose Zone? Here's a diagram that I like to show when talking about the Vadose Zone. It really is just the area beneath the root zone and above the water table. So anything that's unsaturated, the soil and sediments below the root zone would be called a Vadose Zone. And it just means that it's not completely saturated. But if you look at this diagram, you'll see that there's places where we have perched water, that perched water zone where it might be temporarily saturated. And we have quite a bit of complexity, I guess, in water flow, even in a relatively uniform Vadose Zone like the one in this diagram. So here's a map of what we think Nebraska's Vadose Zone looks like. This is actually a map of the depth to the water table across the state, produced by the University Conservation and Survey Division. The light areas, light blue, show areas where it's less than 50 feet to the water table. And we're going to come back to that, but those are areas where we also have a lot of crop production, a lot of irrigation goes on in those areas, and very, very sandy soils. And that tends to mean that we're going to see nitrate in the local groundwater. Well, we've been studying the Vadose Zone for quite a while. This shows a report that was produced back in the mid-1990s where we compared Vadose Zone nitrate to different cropping and irrigation practices. And if we can look at that data over time, it might start to tell us a story about how important the Vadose Zone is in controlling or dealing with groundwater quality issues like nitrate. So I've been involved with a Nebraska Vadose Zone project for the last 3 years. It's actually a website that http nebraskavadose.unl.edu. It has close to 1,200 different cores stored on this interactive GIS map that allows anybody, including you listening to this presentation, to go out and look at what the Vadose Zone, how much nitrate was in the Vadose Zone at different places across the state. So here's an example of from 9 Mile Prairie, that's just outside of the city of Lincoln. It was cored in March of 1991. And what we found is there was very little nitrate beneath 9 Mile Prairie. So we're using this as our control site because there hasn't been any agricultural that we know of on 9 Mile Prairie. This would give us an idea of the background nitrate concentrations. And next to that, we have a calculated pore water nitrate that's in milligrams per liter. We're just using the moisture content from that core and we're converting the soil or sediment nitrate to its milligram per liter equivalent. So you might think of this as being the same milligram per liter nitrate that you might find in a groundwater sample. And you can tell very low concentrations. This is sort of a natural or pristine site that we might compare to any place else in Nebraska. So how do we study the Vadose Zone? Here's a few photographs, including one of my former master's student, Craig Adams. We use a drilling rig, this truck mounted drilling rig on the left-hand side of the screen. They drill into the soil, sometimes up to 100 to 120 feet. And then they bring up sections of core like we're seeing in the center photo, and they're contained in these clear plastic liners. And if you look really closely, you can see that there's a lot of sand in that particular core. That means that the water can easily move through that Vadose Zone at that location. And if it's more or less continuous, that would provide a nice conduit to the water table. So we take a measuring tape after we've collected the core down to the water table, and we can actually measure the depth to water in the same hole that we collected a core from. And so we load all of these core samples into a big styrofoam cooler, and we bring them to the Water Sciences Laboratory. Once we get them to the lab, we'll push them out of the plastic liners, and we'll do a very detailed description of what the sediments look like, what kind of textures they are, if they're sand, silt, or clay. If there's any iron or organic matter in them, we'll note that. And then we'll dry a portion of that sample and extract it and measure nitrate and ammonia in those extracts using this instrument in the bottom of the slide. It's called a flow injection autoanalyzer. So what can we do with all that information? Well, I'm going to give you just a couple of examples. Here's one from the Hastings Wellhead Protection Area. It's a place that we've done a lot of Vadose Zone sampling. We just completed a study in 2019, and we'll be planning to do another one in another couple of years. We sampled actually 32 different locations, but I'm only showing you 6 on this particular slide. And we compared the amount of nitrate that was stored at that same location between 2011 and 2016. And you can see that some of these locations actually we saw a decrease in nitrate, and some we saw an increase in nitrate. So why is that? Well, we think it's because different irrigation and fertilizer management practices lead to leaching in the Vadose Zone. So here's looking at those same coring locations at the two different time snapshots, 2011 and 2016. And we can see for this HC11, there was a big slug of nitrate concentration at about 20 to 22 feet. And when we cored that same location again in 2016, it had disappeared. So that might be kind of surprising. You wouldn't think that nitrate would move that fast, but it actually had traveled all the way down below the 60-foot limit of that particular core sample when it was collected in 2011. If we go over and look at the other core, HC16N, we see that we have an increase in nitrate from about 1.2 to a little over 3 parts per million, micrograms per gram. And we can look at the whole profile and we see that there's at least two to 3 times as much nitrate, pretty much the whole soil profile. So we can relate these changes in concentration to how much irrigation water was used at the surface and how much fertilizer was applied. The two bottom graphs show the moisture content, and we can see that there's a nice correlation between both times that it was sampled, and we think that this moisture content is directly related to the type of sediments that are in the Vadose Zone. So one thing that we've learned from all of this sampling and analysis is that changing from a really water intensive irrigation practice, like it's called gravity or furrow irrigation, to a more water conservative method like a center pivot sprinkler irrigation actually reduces the amount of nitrate that leaches below the root zone. And here's an example where the producer actually changed from furrow irrigation to pivot irrigation between the 2 times that we collected the samples. So this is a great management practice that we can employ that will actually lead to lower nitrate in the Vadose Zone and hopefully over the long term reduce nitrate in our groundwater. In that Hastings study, we also looked at the different types of land use. We cored some lawns in the city of Hastings. We looked at I think 23 different locations where they had pivot irrigation. And then we looked at about 4 to 6 locations that were still using furrow or gravity irrigation. And we just averaged those 3 different types of land use or water use. And we found that residential lawns overall have the lowest nitrate in the Vadose Zone. Center pivot has about twice as much. And furrow irrigation almost always has more nitrate than any other type of land use. So this is something that we're finding in many locations where if we can reduce the amount of nitrate in the Vadose Zone if we would just use less water. Another study that we recently completed is in the Central Platte Natural Resources District. These are 4 different core locations. Some of these are beans, some of them are irrigated, some are not irrigated. I'm not going to go into all the details, but just to show you here in this particular graph or this map that we have a lot of variability, and in many cases we actually have an increase in nitrate in the Vadose Zone. All right, so what does that amount to? This is, we're going to flip our image and look at groundwater now. This is a paper that was published back in 2014 that looked at the changes in groundwater nitrate. You can see these red areas are the places where we see the highest concentrations of groundwater nitrate. This is from 1980 to 1990. This is the Central Platte NRD where we just had those core profiles collected. You can see as we flip through the time snapshots, we go to the 1990s, It looks like we're getting a larger area that's impacted by higher nitrate levels. And then in the 2000 to 2010, even more areas. So this is a very gradual process. Many people haven't even noticed that it's happened, except for people like us, or really that's what our jobs are to study how nitrate gets into our groundwater. So here's a map of the most recent nitrate levels. You see we have the highest levels through the Central Platte, basically along the Platte River Valley, and up in northeast Nebraska, Holt County, Knox County, Antelope County, they tend to have fairly high concentrations of nitrate in the groundwater. And if we compare that to that Nebraska Vadose Zone map, you can see that these areas have a very thin Vadose Zone, also have very sandy soils, and very vulnerable to leaching nitrate from the surface. So I hope that you can appreciate how important the Vadose Zone is for monitoring and understanding how groundwater gets contaminated. Groundwater is especially vulnerable in areas with the sandy soils, high density of irrigation, and shallow water tables. And we've learned over the many decades looking at the Vadose Zone is that it can help provide us with site-specific effects such as changing management, and maybe would help promote the use of more water conservation irrigation methods. And with that, I'll be happy to answer any questions.
We don't have any questions right now. I have no clue what I just did. Yeah.
Why are nitrates bad? Yeah. Good question. They can, well, nitrate in drinking water can, if consumed by infants especially, can be converted into nitrite. And then nitrite can result in a medical condition called blue baby syndrome or methemoglobinemia. So that's one of the human health effects from nitrate in drinking water. More recently, we've learned that there's certain types of cancers that are associated with drinking water that has high concentrations of nitrate. So it's basically that it's a reactive ion, and that it can interfere with many of our medical processes, many human health processes. Okay, did I say uranium is reacting with nitrates? Yes. We think that nitrate leaching from crops and intensive use of irrigation water is leading to higher concentrations of uranium in groundwater. Basically, you might think of it as a way of accelerating weathering of soils and sediments, and that weathering process is actually promoting leaching of uranium and in some cases selenium, other trace elements that occur naturally in soils. Can you have a private well tested for nitrates? Absolutely. There are many laboratories in the state that do nitrate testing, including the State Health Lab here in Lincoln, but nitrate is probably the most requested test for private well water quality. And here's one: has the 2019 flooding caused any increase in nitrates? Or I think it's, yeah, is it too soon to tell? I think it is too soon to tell. But that's a very good observation. I would think that in some cases we might see an increase in nitrate due to flushing of the Vadose Zone. And how long does it take for existing nitrates to disappear from the Vadose Zone or groundwater? Well, you know, it's taken decades to reach the level that we are now. I think it's going to take decades to have nitrate concentrations decline. So it is a very long, drawn out process for nitrate to dissipate. All right, source of nitrate. Types of nitrogen fertilizers, manure applications, or other factors have an impact, or is all nutrient supply the same? Well, you know, if all factors are equal, inorganic sources tend to be the most highly leachable. Livestock manure in some cases leads to excess nitrogen in soil. And if you have excess nitrogen of any form, in excess of the crop needs, it will leach below the root zone. But inorganic nitrogen fertilizers are probably by far and above the most common cause of nitrate contamination in groundwater. Okay, a question from West Point, Cumming County. There is a large cattle feeder area. There's definitely a lot of animal feeding operations in Cumming County. How close do you take samples from the cattle fields? I know local entity has a lot of pushback from cattle feeders, crop farmers in our area. Would you like to comment on this? Well, you know, Nebraska, most of Nebraska land is private, under private ownership. That makes it, in some cases, difficult to get samples of the groundwater beneath private land. I can see instances where we have lots of cooperation from producers. They want to know if things are getting worse or getting better under their property. But there may be cases where someone's not sure if what they're doing is leading to unintended consequences. So I think they might be reluctant because they just don't really understand how nitrate gets into our groundwater and what we might be able to do about it. Good questions. All right. Well, I think we might move on to our next presentation then.
Wonderful. Thank you, Dr. Snow. Good evening, everyone. My name is Alison Klein, and I am a high school science teacher at Kearney High School in Kearney, Nebraska. And I'm excited to be with you guys this evening to talk to you about the Know Your Well program and the partnerships that we have made for water education. I was introduced to the Know Your Well program when I participated in a UNL workshop back in the summer of 2018. The UNL workshop was hosted called Water Education Leaders for Secondary Science program, or the WELLS 2 program. And while participating in the WELLS 2 program, some graduate students and Dr. Snow came in to talk to us about the Know Your Well project. I was really interested in learning more about how to get my kids active in the field, collecting and working with original data and using protocols that have been approved by scientists. And while we were there during those 4 days, we were able to go to East Campus, actually see the technology and the equipment that they used. And it seemed very approachable and something that I felt like I could reproduce in my classroom. After we participated that summer, we had the opportunity to sign up. And if our site was selected, then UNL teams came to our site and talked to our kids about how to monitor water quality and the resources that we use to know more about the water beneath the soil. So, and we know that as a teacher, professional development empowers us to be more effective in the classroom. And so as a result of participating in the WELLS program and the Know Your Well program specifically, I've learned how to access well information from government websites like the nebraska.gov website for groundwater that has interactive maps that shows you well data for your region. And not only find and access the data, but understand how to interpret the well data that it contains. That's a really fabulous experience to take the kids just to Kearney, Nebraska, and to see all of the dots show up for the different types of wells and the way that we interact with water, even within a municipal area like Kearney, Nebraska. As a teacher in the Know Your Well program, we learned about describing well locations and also protocols for collecting water samples so that our data could be repeatable. It was great to see kids learn how to organize materials, to work in the field, and to take qualitative and quantitative water samples. As you can imagine, whether you're in a small school that's a Class D and you maybe have a class of 20 kids, or you're in a Class A school with 1,400 students in grades 9 through 12, there are challenges to implementing anything. As we continue to move forward, now that we're ending the 2021 school year, we still do use many of the Know Your Well protocols that were introduced back 2 years ago when we started working with the project. Some of the things that I would do differently, and I feel would work well with any school, is that I would use a well on your school site to train your kids on how to describe a wellhead and to collect data. And then I would organize small groups to go out and collect water from the private well sites. Logistically, it's a lot easier to take maybe a small FFA group of students that are interested, or a science club, or an after-school program. Having a team of 5 to 10 kids would be easier to transport and easier to manage when you're in the field working. We had many people around the community that had private wells that wanted to participate in the program. So having the ability to take those small groups to those sites, I feel would work really well. Lower transportation fees are also definitely a perk. And then I would bring that water data back, and you can analyze the water samples in a large classroom lab setting. It's easy to have station rotation where students test the same test. So the well program provided kits for common ions that are monitored in drinking water and groundwater. So you could have one team that was focused on just testing for iron, and another team focused on just testing for copper. And you could have a third team looking at nitrates. So that would be one way to scaffold the lab experience in the classroom. The Know Your Well project was really a fabulous program to become part of. In 2019, it was right when Kearney High School also introduced our FFA program, and it sparked so much interest within our community for water education. So one of the side effects of participating in the Know Your Well program is that we also worked with the city of Kearney in educating the community on the role of rain gardens and different landscaping techniques. So we helped install and design a rain garden, an education rain garden that's near Kearney High School, right by STEINBRINK landscaping. So I have some pictures here of our installation day that we did with a summer group and some of the partners that helped make this project possible. And you can see on the left the picture after we were done with installation. Not only did we include students in communicating to the school body about the importance of slowing down rainwater runoff to increase its quality through different techniques, rain gardens being one of them. We also worked with the students in our Bearcat design project, and their focus in Bearcat design is to do media work for us. So the sign that is posted at the site was designed by a student at Kearney High School as well through our Bearcat design project. So it's great to see not only science and environmental education, but to see those also working with the arts as well. In addition to partnerships with the city of Kearney and STEINBRINK Landscaping and Nebraska Extension on our rain garden, we've also worked with the University of Nebraska Kearney and the Audubon Society at our outdoor learning area, which is also part of the Kearney property. And our focus at the outdoor learning area is on water quality observation and the role of wetlands. And we partner with the Kearney High School FFA program in supervised agricultural experiences, where I'm using some of the same protocols and ideas and ion monitoring that we did in the Know Your Well program to monitor the water quality for Turkey Creek and the water runoff from our geothermal heating system and our parking lots that you can see retained here in this retention pond. So it might have just started with, I wonder what's in my water from the Know Your Well program, then went to inspiring kids to participate in the rain garden program, inspiring kids to want to do extracurricular projects related to water quality through FFA. And the last partnership that we are working with at Kearney High for water quality education is monitoring, witnessing watersheds. And this is a project with UNK and Dr. Mary Harner. And Dr. Mary Harner works with the Platte Basin time-lapse project. And we installed a time-lapse camera and an audio recorder at COLA. And it takes images of the same section of our restored prairie every day. And we have them, the first ones were taken in 2018. And they are stored in a drive, and we have students working in Bearcat Design Project to create images and to communicate how watersheds change over time and the role that they have in healthy ecosystems. So what started off as a really great summer opportunity to learn more about water, to be a better educator, has blossomed into multiple projects and partnerships that have impacted students, not only through the Know Your Well project, but also through partnerships with the University of Nebraska and the city of Kearney.
All right, any questions for Alison? All right, I think it's really cool what you did there with the students. So, not seeing any questions though, so it's all right. Oh, here we got one. They're asking what types of technology do the students use for those projects?
Okay. Great question, Cece. Thanks for asking. I'm guessing that Dr. Snow and Tania will also talk about the type of technology that we use when we do water quality measurements in the lab. The equipment that was provided for students is designed to give us qualitative, so answering the question of is the ion present, and basic quantitative measurements to tell us ranges of the concentration. The goal was that if we could train students in protocols that would be repeatable, that they could also get high quality research data out of those measurements. And then when we would compare our data, there should be correlation with what the lab was finding at the University of Nebraska. So the kits are really great because each of the kit was packaged in its own box, and it would say a protocol list, like this is how you perform this test, and it would walk the kids through it. And most of them have color indicators where it would change in the color that you would see from maybe a light orange to a dark orange, and it would have a calibrated range. So you could hold your sample up next to it to say what your parts per million or parts per billion count was. Yeah,
I have this question here. Has this education program been encouraged in all Nebraska high school FFAs? I know you're not an FFA person, so I don't know if you know what all the...
That's a great question. I don't know that many, I don't know if it's something that many FFAs have done across the state, but the supervised agricultural experience of water quality monitoring is a nationwide project any student can participate in.
And Dr. Snow did put a link to the drinking water testing in the chat for if anyone wants to grab that link there. All right, I think we can move on to our next speaker.
But before we move on, I just wanted to add, we're actually planning to grow Know Your Well. So we really appreciate Alison's presentation. We've had a outpouring of support from Nebraska teachers on this project. So I'm really excited about expanding the number of schools that will participate, and just look for that opportunities in the near future for getting more schools involved.
All right, well, we can move on to our next speaker. That would be Jesse with if you're ready.
Thanks, Eric. Tania, do you have slides that you wanted to present, or should we just save those for later?
We can do the video.
Okay. So I'll give a 30-second preamble to this video. We just recorded this about 3 weeks ago, and this is a quick around the lab in 9-minute tour. I think it'll give you a great initial taste of, when people wonder, how do they know what's in the water, which is a great question to ask. It's because of laboratories like the Water Sciences Lab and some of the really incredible cutting edge equipment and techniques and people that the lab employs. So with that, I will queue up this tour and optimize it for video.
And welcome to the Water Sciences Laboratory on the University of Nebraska East Campus. I am Dr. Saptashati Tania Biswas, the Research Laboratory Manager here. We are a university research core facility, part of the Nebraska Water Center and the Doherty Water for Food Global Institute. The lab has been operating for over 30 years, specializing in analytical services and training to support university students, water researchers, and stakeholders. Whether you want to know what's in your water in Nebraska or in Nepal, we have the equipment and expertise for you. The lab has analyzed thousands of water, soil, and biological samples in support of water resource management and science. Ultimately, the lab helps to build a data-driven picture of Nebraska's water quality, identifies where remediation is needed, and equips the next generation to tackle those challenges. Our facility, which is right behind me, is around 4,000 square feet and houses over $3 million in high technology lab equipment. Let's visit our main sample preparation laboratory that also houses our standard method instruments. Many of these methods are used in research and monitoring projects, such as the Nebraska Vadose Zone monitoring and the Know Your Well groundwater quality monitoring programs. This instrument is called an autoanalyzer and is used to measure concentrations of dissolved chemicals such as phosphate, nitrate, and ammonia. We run these tests on several thousand samples each year. The whole process is automated for rapid, precise, low-cost analysis. Chemical reagents are added automatically to samples and form uniquely colored complexes. The instrument measures the intensity or absorbance of the color to quantify how much is present. We use a similar chemistry in our flow injection autoanalyzer, also known as the latchet, for measuring nitrate and ammonia in soil extracts. This is one of our ion chromatographs where we use a column to separate and measure anions such as chloride, bromides, sulfate, fluorides in water and extracts. We also offer specialized methods. For example, we recently developed a method for measuring chloride and chlorination residues in spices such as black pepper and thyme. Most people don't realize that chlorination is used in water treatment for food production and that byproducts can be left behind after processing. This is the organic carbon analyzer and used for measuring total and dissolved organic carbon in water by persulfate oxidation method. The organic carbon in the acidified sample is oxidized to carbon dioxide and measured by the infrared detector. Organic carbon concentrations is a good way to measure water purity and safety. We have other instruments like the atomic absorption spectrophotometer that we use to measure cations in water like sodium, potassium, magnesium, and calcium. We will soon replace this AA with an ICP spectrophotometer that enables simultaneous analysis of a wider range of elements. We already have an advanced version for measuring very low concentrations of metal and other trace elements. This is the inductively coupled plasma mass spectrometer, or ICP-MS, that is used for measuring very low concentrations of elements like arsenic, selenium, uranium, lead, cadmium, manganese, and so many others. The sample is aerosolized and introduced into the plasma chamber. This enables the ICP-MS to measure the formation of charged ions through kinetic energy discrimination that removes interferences and makes it highly sensitive. The ICP-MS can operate in tandem with another ion chromatograph system to analyze a variety of metal species. Additionally, we have developed a multi-residue method to measure polar pesticides, such as glyphosate, which is the main chemical in Roundup pesticide. One of our core analytical service areas is environmental mass spectrometry. This technology measures a variety of pesticides like atrazine and newer chemicals like neonicotinoids, as well as human and veterinary, pharmaceutical, and other organic chemical pollutants. These advanced instruments are very sensitive and can quantify these chemicals at parts per trillion level, something not possible with other instruments. The two here are the liquid triple quadruple mass spectrometer and gas chromatograph mass spectrometer, each tailored to certain types of chemicals. Installed in 2019, the Xevo TQ by Waters is our latest instrument that is 1,000 times more sensitive than our older LCMS. This has doubled our throughput while increasing confidence in detection and accuracy of contaminants of emerging concern. Our final core analytical area is the stable isotope analysis. Stable isotope analysis measures isotopes of hydrogen, carbon, nitrogen, and oxygen at very high precision. Natural variations of the abundance of these isotopes can help discover the origin and flow of these elements in the environment. Stable isotope analysis often fingerprints sources of contaminants such as nitrate and can also be used in studying chemical reactions and metabolic processes. The Water Sciences Laboratory offers methods for stable isotope analysis of gases, water, and solids. The lab is well known for its methods for detection of isotopic composition of nitrate and ammonia, nitrogen, to support the investigation of nitrate contamination of groundwater and environmental fate and transportation. In the past few years, we have trained an average of 25 to 35 students and researchers annually. They all start in this facility. We offer several training modules on good housekeeping and laboratory practices, use of basic equipment such as analytical balances and micropipettes, sample preparation techniques, and advanced analytical methods involving instrumentation. The high quality results start with good laboratory practices. Maybe you would like to be a water scientist. I hope you have learned a little bit today about how we analyze the water around us to help students, researchers, and water professionals make decisions related to the incredibly important natural resource. I invite you to learn more about the lab and our services at watercenter.unl.edu/WSL. Thank you for joining me.
Tania, I don't know what's left to say after that.
I know, Jesse, and I should thank you for being our videographer and director for this video.
Well, thank you. Apologies for that. I had another video that started playing in the background. Yeah, I just wanted to put in a really quick plug for other follow-up that people may have at that same website, watercenter.unl.edu. We have a lot of publications for lay people. If you're a non-scientist or you don't have a technical background, but you just want to learn more about water and water analysis, we have a quarterly newsletter that comes out, and we did a whole edition on the Water Sciences Lab that came out about a year ago that has a bunch of good information. So that's, I'll throw the link in the chat for folks who want to follow up that way.
Looks like there's a question probably to Tania.
Yeah, I do see the question. I'm curious, how do you know when to make a new water test? It depends who you are. We get a variety of samples from researchers who are working on different research projects, and they have different hypotheses. So, you know, they would send us samples when they want to do a reading for whatever projects they are working on. We get samples from different monitoring projects. And then the natural resource division. So it depends upon what they're working on and when they feel they need to do the test. We do seasonal tests during the farming season. So before starting off the planting, there'll be a whole bunch of samples coming in, and then post-harvest. So people have a variety of reasons for testing water. Testing is important. Don't know if I answered your question, but happy to take a follow-up. Dan, would you like to say something?
Yeah, I think they're asking, when do we develop a new method?
When we are asked to develop a new method, we have routine testing that we do in the lab for standard methods that are already developed, and a variety of labs offer those methods. It's mostly the emerging chemicals, also called the emerging contaminants, and various species of different minerals. So those are, there are newer chemicals that we test in water and soil and other environmental matrices. So whenever we get a request for such chemicals to be tested, then we first have to develop a method if it has not been part of a routine analysis before. Do you think I've answered the question, Dan?
Yeah, I could give an example. We started testing for neonicotinoid insecticides several years ago because a researcher was interested in these new insecticides in surface water. And there's been an explosion in the requests for testing for these new insecticides. Some of you may have heard of the Alt-N energy, bioenergy site where there were found to be fairly high concentrations of these insecticides. So we ended up expanding our method to include even more insecticides, partly because of this event that has occurred, and try to understand the extent of the contamination and whether water supplies might be vulnerable to contamination from these chemicals.
Thank you, Dan. Anne has a question in the chat box. Are there water issues that you will be studying with water treatment plants, etc., dealing with the COVID virus, since it is showing up? Maybe this is not your area since your water issues are more groundwater related. I know this is a strange question, and it is not a strange question. It's a very relevant question, right? We are in a pandemic and meeting on Zoom. So it makes sense that there will be a question on COVID testing. We have tested water samples from treatment plants, but not COVID or any viruses and bacteria. Our scope of services is chemical testing. We do some immunological assays for microcystin, and that's about it for the kind of biological tests in the biological realm. But I know there are a lot of research going on in civil environmental engineering, where they are doing tests for the COVID virus and seeing that if there are residues from the sewage treatment plant. So this is a good question. Dan? This answers it?
Yeah, I would only add to that. Yeah, we have written proposals to test for COVID viruses in wastewater, along with chemical contaminants, some of the emerging contaminants that Tania mentioned. We can use those as tracers of wastewater inputs, say into surface water, and relate that to the potential for COVID virus or other virus contamination of surface water.
Thank you, Dan. Anne has another question. Where does our funding come from for the lab?
It comes from a variety of sources. We get some support from the Nebraska Research Initiative. We also receive contributions from the University of Nebraska colleges, such as the Institute of Agriculture and Natural Resources and College of Engineering, as well as Arts and Sciences. And then finally, we also charge service fees for all of our tests. And we use that as part of our income to support our staff and our student interns.
Yep. Thank you, Dan. We have a couple of questions in the Q&A box. The first one is there one base test, like doing some kind of spectrography? We do screening with spectroscopy. Don't know if that answers the question. But our tests are multi-residue, so there will be, so with one test, depending upon what kind of spectroscopy method we use, or by spectroscopy, it will be mass spectroscopy for trace level analysis, whether it's organics or inorganics. So we can, with one test, we can measure many of those analytes, group of compounds. So yes, it's we develop multi-residue methods. That makes it more time efficient, cost efficient. Okay, let's get another question. Dan, you should answer this. So did you find neonicotinoids?
Yes, actually, we have detected neonics in the surface water samples, and even in some of the air samples that were collected. It's pretty clear that there's going to be residues of insecticides in that area for quite some time.
Couple of more questions. Deborah has a question. Do you use this information to recommend and advocate for policies that will protect our water sources?
Yes, anytime that we can use research results to support new practices. For example, the research in the Vadose Zone that showed use of pivot irrigation over gravity irrigation, that would be a very useful policy to develop in helping to protect local groundwater from contamination from nitrate.
Yes, the answer is yes, as Dan said. Becky has a question. Are you testing oil and gas production sites?
I can't say that we have. There was several years ago a lot of interest in hydrofracking and the potential for fracking to contaminate local groundwater. We don't have a lot of oil and gas production in Nebraska, so that it wasn't a big issue for our state. I know other states like Wyoming and Pennsylvania, it was much more prevalent.
Thank you. Deborah's next question is, do you think climate change is impacting our water resources in Nebraska?
Absolutely. Climate change is affecting all of our natural resources. In some ways, it maybe would improve growing where we might have more water, but we could also have incidences of flooding. Flooding in general can lead to water pollution, both surface and groundwater. So climate change definitely will affect our water. No question about it.
And also heavier precipitation, and that leads to a lot of runoff, so more opportunities for all the crop protection chemicals to flow from the field into the surface water.
All right, we have time for a couple more questions, and then we'll have to let this go for tonight. But
so the next question, and let me know, Eric, when to stop, because I do see a list of questions. So let me know.
We'll answer these next 3 questions, and then we'll go on to the end of the program.
Okay. Charlene has a question. Which cancers have been linked to high nitrates in the water?
I believe one of the cancers is non-Hodgkin's lymphoma. Martha Rhodes has done a lot of research in that. She's a faculty member in the School of Natural Resources. And if I recall correctly, non-Hodgkin's lymphoma was one of the cancers that was associated with the occurrence of nitrate.
Anne has a question. Is GC Resolve connected to UNL water testing? Jesse, yes, please. Go ahead.
I can take that one. I just talked to Graham Christensen yesterday. He and his organization has been involved with a Professor Shannon Bartelt Hunt in civil engineering. She has a really nice program for water quality and citizen science testing for surface and groundwater across Nebraska. And I'll try to track down a link for that. But yes, GC Resolve has been involved in helping to perform that water testing.
Thank you, Jesse. Final question from Vern. Are you in favor of stopping water fluoridation?
Who's going to answer that one?
We test samples. That's high quality control. We'll find if there's fluoride for you.
The only time fluoride is really a problem if there's too much of it in the water. So I think we all need a little bit. And the only time it's a problem if we get too much of it.
Thank you, Dan. And I know some of you had other questions, but we possibly couldn't answer all of them in this time. But go to our website and email us. We want to hear from you. So please email us with your questions.
All right. Thank you guys for all attending. Thank you guys for your expertise in the water issues surrounding our state and the quality work that you guys do at the UNL there. Thank you very much for your time. There's a quick survey that will be right now. So just answer that, and thank you guys for attending.